My project is on analyzing the expression of high mannose and glycans in breast, ovarian, and colorectal cancer. Three of the leading causes of death due to cancer in the U.S. are colorectal, breast, and ovarian cancer. Colorectal cancer is the second most common cancer and the third most common cause of death in the U.S. Its mortality rate is 900,000 deaths annually. The diagram on the right shows how the tumor progresses. When the cancer reaches stage 3, it is spread to one or more lymph nodes in the area, and by stage 4, the cancer has metastasized to distant sites, such as the liver, lungs, or bones. Similar patterns can be found for the stages of the other two cancers. As these cancers are often asymptomatic in the first years, it is imperative to develop sensitive diagnostic tools and potential therapeutic targets. Glycans may offer an answer. Glycosylation, one of the most common post-translational modifications, refers to a reaction in which carbohydrate residuals, or glycans, are attached to a non-carbohydrate entity. There are two types of glycans, N-linked and O-linked. This project focuses on high mannose and glycans. I will be investigating how high mannose and glycans are expressed and their role in colorectal, ovarian, and breast cancer. First, the cells or tissues are homogenized and membrane proteins are extracted from it. Further, glycoproteins may be enriched to access lower abundance proteins before subjecting them to enzymatic or chemical release of N-glycans from the protein backbone. The released N-glycans can now be analyzed using mass spectrometry combined with separation methods such as liquid chromatography, which is one of the most widely used hyphenated techniques since it requires a small sample size and offers accurate analysis of glycan structures with high sensitivity. This is a summary of the findings from colorectal cancer papers. As you can see, all of them show an increase in high mannose and glycans. I found that some of the studies mention where they got their sample from, and they are from different parts of the world. For example, the third study here is from Netherlands. These are the high mannose structures found in colorectal cancer used in one of the studies. The graph on the left shows an increase of high mannose and glycans from tumor versus normal tissues for individual patients, and the graph on the right shows an average relative abundance with standard deviation. This is a summary of the findings from the ovarian cancer papers. Again, all of them show an increase in high mannose and glycans. Similarly, these graphs show an increase in relative intensity percentages of high mannose structures in tumor versus normal cell lines for ovarian cancer. This is a summary of the findings from breast cancer papers. Like the others, all of them show an increase in high mannose and glycans, and the second study here obtained samples from China, and the third from South Korea. I mention this because this increase in high mannose and glycans appears across many different ethnic groups. All the studies for all three cancers either use cell lines or tissue samples from humans, with the exception of one of the breast cancer studies, which also profiled mouse serum in addition to human serum glycoproteins of breast cancer patients and controls. Using data from our breast cancer study with patients in China, the ratio of relative intensities of high mannose structures in cancers to normal tissues were all greater than 1, showing an increase in high mannose and glycans. Not only are high mannose and glycans significantly increased in relative intensities, but also complex and glycans show a decrease in relative intensities. These graphs also show that high mannose and glycans increased in relative abundance, while complex and glycans decreased in both colorectal and ovarian cancer, which is consistent with the claim. A survey of journal papers concurred that high mannose and glycans were elevated for colorectal, ovarian, and breast cancer. Consistent with the literature review, the analysis from publicly available datasets also showed a significant increase in the relative abundance of high mannose in tumor tissues and cell lines compared to non-tumor ones. Combined with the findings of the decrease in complex and glycans in these studies, the rise of high mannose and glycans may be explained by a truncated glycosylation process. This would be due to the shorter replication time of tumor cells. The increase in relative abundance of high mannose and glycans being a commonality between all three cancers can serve as an indicator for tumor presence. It is interesting to note that this increase in high mannose structures, coupled with the decrease in complex structures, appeared in tumor samples from different ethnicities.
Question would be, did you see in those references if the surface protein glycosylation was more important than the bulk glycosylation or cytoplasmic proteins glycosylation? Did they mention anything about surface protein?
That wasn't really in the scope of my project, but that sounds like a really interesting thing to look into, and I'll be sure to look into that later.
Okay, second, a little bit connected question is that do you explain somehow what is the impact of those monosylation of this monosylation of the surface proteins on the cancer progression? How do you imagine for yourself what is the mechanism of impact of this monosylation?
Sorry, can you repeat the question again?
Yes. So why monosylation is important for cancer progression?
Oh, okay. So are you asking like how is it related to cancer progression, the high mannose and glycans?
Mhm.
Okay, so like I mostly looked into how it increased in high mannose and glycosylation, and in the glycosylation process, um, the high mannose and glycans occur are created first. So because in cancer the tumor cells would be dividing rapidly, and there might be a truncation of the glycosylation process, I was thinking that um, that would be why there would be more abundance of high mannose and glycans compared to the other types of glycans.
And in the end, I would probably add that in my experience, in my research, I was working on the mannose receptor of the macrophages, and this receptor can induce some kind of immunosuppressive downstream pathways inside the macrophages and probably other myeloid cells. So maybe it's explained by the modulation of immune response in the tumor because mannose receptor is highly expressed on the macrophages. It may be one of the mechanisms, but
Yeah,
thank
no,
you, great research.
So I'm a 10th grader in high school, and so I didn't have any previous knowledge on glycobiology at the start of this program, but Dr. was very helpful, and she would explain the necessary concepts and teach me how to do my own literature search and create a presentation and answer my questions by email. And it was really interesting to analyze the publicly available data and generate graphs. And I even got to the chance to explore platforms used by researchers in this field, such as Glycobench, Espacy, and Unicarb databases. So overall, it was a really great learning experience. So thank you, Dr. SkoolMentor.